Have you ever stopped to wonder how we peer into the incredibly tiny, dynamic world humming inside our own cells? How do scientists actually glimpse the intricate structures and processes that animate life, far beyond anything our naked eye could ever hope to perceive?
It's a fascinating question, isn't it?
Yeah.
And it's one that's driven science for centuries, really.
Yeah.
Constantly pushing the boundaries of what we can actually see.
Yeah, and the answers have, well, they've completely revolutionized biology.
Absolutely.
And that's pretty much our mission today on the deep dive. We're going to plunge into the absolutely fascinating world of visualizing cells and molecules.
Right.
Our journey today unpacks a foundational chapter from Molecular Biology of the Cell, 7th edition, revealing the groundbreaking techniques used to observe this microscopic universe within us.
And the idea is, by the end of this deep dive, you'll have a real shortcut, a good handle on cellular imaging
Right.
from its very humble beginnings.
All the way to the cutting edge stuff that can literally pinpoint individual molecules.
Exactly.
Okay.
We're drawing exclusively from this textbook, which is, well, it's a cornerstone of the field, so you can expect clear explanations, hopefully, and a look at some truly ingenious experimental designs, plus how these tools are actually used in, real biological research.
And we'll keep coming back to this idea of trade-offs, right? Because it seems like every imaging choice involves some kind of compromise.
That's a core theme, definitely. It's never just a simple upgrade. There's always a fascinating balance to consider. It makes you think about why a researcher chose a specific method.
Okay, let's unpack this. Cell biology, as a field, it really kicked off back in the early 19th century.
Right, when light microscopes got significantly better.
And this new ability to see things led Matthias Schleiden and Theodor Schwann to propose the cell doctrine in 1838.
A landmark moment, basically saying, hey, all living things are made of cells.
But there was a huge challenge right away.
The scale problem. I mean, think about it. A typical animal cell is only about 10 to 20 micrometers across. That's what a fraction of a human hair's width.
Tiny. Far too small for the naked eye.
And on top of that, most cells are basically colorless and translucent. It's like trying to see details in clear jello.
Yeah. So early microscopists had to get clever, using stains, right, to add some contrast.
Exactly. Anything to make things stand out.
And that challenge of scale, it's still central today. I mean, the textbook has this great figure starting with a thumb.
Ah, yes, figure 9 to 1.
And zooming in factor by factor. You quickly go from what your eye sees to needing a light microscope for bacteria.
And then needing an electron microscope for viruses or ribosomes, those tiny protein factories.
What's really amazing now is how these new super-resolution techniques are bridging that gap, letting us see things with light that were previously impossible.
It's pushing light microscopy into territory once reserved only for electron microscopes. It's pretty exciting.
So speaking of conventional light microscopes, they hit this fundamental wall for, over a century.
The diffraction limit.
Yeah.
It's tied to the very nature of light itself. Well, visible light behaves like a wave, right? It has a wavelength between about 0.4 and 0.7 micrometers. And you just can't resolve or distinguish two objects that are closer together than about half the wavelength of the light you're using.
So around 0.2 micrometers or 200 nanometers, roughly.
Exactly. That's the theoretical limit for conventional light microscopy. So fine details inside bacteria or the internal folds of mitochondria, they just blur together.
Okay, so if light's a wave, how does that actually cause blurring? Is it like overlapping waves canceling each other out?
That's pretty much it. It's called diffraction and interference.
Sure.
When light waves from a point source pass through the lens, they spread out and interfere with each other. This makes what should be a perfect point appear as a blurred disk, often with faint rings around it. That's the airy disk or the point spread function.
Right.
And when the blurry disks from two nearby points overlap too much, your eye, or the detector, can't tell them apart anymore. They merge into one blob.
So it's not just about having a perfect lens, it's physics.
It's fundamental physics, yeah.
Yeah.
And that ability to tell two close objects apart, that's what we call resolution.
Okay.
The key factors are the wavelength of light shorter is better, so blue light gives slightly better resolution than red.
Right.
And the light gathering ability of the lens, which we call its numerical aperture. A higher numerical aperture means it collects more light angles, leading to better resolution.
So best case is about 0.2 micrometers?
Under ideal conditions, yes. But it's crucial to distinguish resolution from detection.
What's the difference?
Resolution is telling two things apart. Detection is just seeing one thing. So you can actually detect a single fluorescent molecule or a very thin structure like a microtubule, even if it's much smaller than 200 nanometers.
Ah, okay. You just can't see its true tiny width. It still looks like a blurred spot.
Exactly. You see a blurred spot of light, but you know something's there. You just can't resolve its actual shape or separate it from another one right next to it.
Got it. Is there anything else limiting what we see, especially with faint signals?
Oh, yeah, definitely. Photon noise.
Photon noise.
Think of images as being made of particles, photons. Detecting them is a bit random. It's statistical. If the light is dim or you take a really short exposure, you get these random fluctuations in the number of photons hitting each spot on your detector. It looks kind of speckled or noisy.
Like static.
A bit like static, yeah. This noise can obscure faint details. The more photons you collect, a brighter signal or longer exposure, the better your signal-to-noise ratio and the clearer the image. It's a huge deal for fluorescence microscopy, where signals can be really weak.
Okay, so if fixing and staining cells can mess them up, lose components even, how do we look at them alive? That seems critical for, seeing processes happen.
It is absolutely critical. And that led to developing contrast-enhancing techniques that work on unstained living cells. Beyond just basic bright field, where light shines straight through, things like dark field microscopy, you shine light from the side, an oblique angle. Only the light that scatters off structures in the cell enters the lens. So you see bright objects on a dark background. It can be quite dramatic.
Cool effect. What else?
Then you have phase contrast and DIC, that's differential interference contrast. These are really clever.
How do they work?
They exploit tiny differences in refractive index within the cell. Basically, as light passes through denser parts like the nucleus, it slows down slightly compared to light passing through the watery cytoplasm.
Ah, like you're running through mud analogy earlier.
Exactly. It causes a subtle shift in the phase of the light waves. Our eyes can't see phase shifts, but these microscopes convert those invisible phase differences into visible differences in amplitude or brightness.
So suddenly, transparent things become visible. You see edges, organelles.
Precisely. You get contrast in living, unstained cells, allowing you to see their structure and, importantly, watch them move and change.
Which is perfect for things like watching cell division, mitosis, or cells migrating.
Absolutely. And often, because these processes are slow, researchers use time-lapse microscopy. Take a picture every few minutes, then play it back fast.
Like a biological fast-forward, revealing things that happen over hours or days.
Yeah, it brings those slow, dynamic processes to life.
Now, how have computers and digital cameras changed the game? Because our eyes have limits, right? Especially with dim light or subtle contrast.
Oh, digital imaging has been transformative. It overcomes imperfections in the optics, yes, but more importantly, it blows past the limitations of the human eye.
So?
Our eyes aren't great in dim light, and we can't easily discern small brightness differences on a bright background. But modern digital cameras, often using CMOS sensors like in smartphones, are incredibly sensitive. They can literally count individual photons. It's better than our eyes.
Wow.
This means you can image living cells for long periods using very low light levels, which minimizes damage to the cells. Plus, the images are digital files.
So you can process them.
Exactly. You can use software to correct for lens flaws, enhance contrast mathematically, subtract uneven background illumination. You can pull out details, even in seemingly transparent objects that were totally invisible before.
Okay, but what about thicker samples, like a piece of tissue, not just cells in a dish? You can't just shine light through that easily, can you?
No, you generally can't get high resolution through thick tissue. So that forces another approach, sectioning.
Cutting it into thin slices.
Very thin slices, usually 0.5 to 10 micrometers thick. But first, you have to preserve the tissue structure.
Fixation, right, using chemicals.
Yes, typically chemical fixation with something like glutaraldehyde. It cross-links proteins, essentially locking everything in place.
Then what? The tissue is still soft.
Right, so after fixation, you dehydrate it and embed it in a supporting medium, like paraffin wax or plastic resin that hardens. Now you have a solid block.
Ready for slicing.
Ready for the microtome. It's like a very, very precise deli slicer using a super sharp blade, often steel or even glass, to cut those thin sections.
And then you put those on a slide and stain them.
Exactly.
Yeah.
You can use traditional stains like hematoxylin and eosin H&E, which give you general information about cell structures based on charge. Hematoxylin stains acidic things like DNA blue-purple. Eosin stains basic things like cytoplasm pink.
The classic purple and pink histology look.
That's the one. But for pinpointing specific molecules, you really need something more targeted, like fluorescent probes.
Which brings us to fluorescence microscopy. This seems like a really powerful tool.
It is incredibly powerful. The basic idea is simple elegance. Fluorescent molecules, or fluorophores, absorb light energy at one wavelength,
Like blue light, say.
and then very quickly re-emit it at a longer wavelength.
Like green light.
Exactly. So you shine the excitation light, blue, on your sample, and then you use a special filter to only look at the emitted light, green.
And your labeled molecule just glows against a dark background.
Precisely. It makes it possible to detect very small amounts of a specific molecule because the signal is so distinct from the background.
What does the microscope setup involve?
It's similar to a standard light microscope, but with a few key additions. A powerful light source, like a laser or arc lamp, two sets of filters, an excitation filter that selects the wavelength hitting the sample, and an emission filter that selects the wavelengths reaching the detector, and usually a dichroic mirror.
What does the mirror do?
The dichroic mirror is clever. It reflects the shorter excitation wavelength towards the sample, but allows the longer emission wavelength to pass through towards the detector. It efficiently separates the excitation and emission light paths.
Okay, so what can you do with this?
Tons of things. One technique is in situ hybridization, or ISH. You use fluorescently labeled probes, usually short pieces of DNA or RNA, that are designed to bind specifically to certain RNA molecules within the cell or tissue. It lets you literally see where specific genes are being expressed.
You can map out gene activity? Cool. What about proteins?
For proteins, the workhorse is immunofluorescence. This uses antibodies.
Which are proteins our immune system makes to target specific things, right? Highly specific.
Incredibly specific. Each antibody recognizes a particular target molecule, its antigen. So you can generate antibodies against almost any protein you're interested in.
And then you stick a fluorescent dye onto the antibody.
Exactly. You chemically link a fluorescent dye, like fluorescein green or rhodamine red, to the antibody. Then you apply this labeled antibody to your fixed cells or tissues. It binds only where its target protein is located.
And you can use different colored dyes for different proteins in the same cell.
Yes, that's a huge advantage. You can label protein A green and protein B red and see if they are in the same location or different compartments. It's fantastic for comparing distributions.
Is the signal always strong enough?
Not always, especially if the target protein isn't very abundant. So often researchers use indirect immunocytochemistry.
How does that work?
You use two antibodies. First, an unlabeled primary antibody made in, say, a rabbit that binds to your protein of interest. Then you add a secondary antibody that's labeled with a fluorescent dye and is designed to recognize any antibody made in a rabbit.
Ah, so multiple secondary antibodies can bind to each primary antibody.
Exactly, you get signal amplification. Many fluorescent tags bounding indirectly to your target, making the signal much brighter and easier to detect. We see this used beautifully to visualize things like the microtubule cytoskeleton.
But there's that trade-off again, isn't there? You're only seeing the labeled bits. The rest of the cell is invisible.
That's a key limitation of fixation and staining, yes. You lose the dynamic context, and you only see what you specifically labeled, which leads us to GFP.
Green fluorescent protein. The jellyfish protein.
The game changer. Discovered in the jellyfish Aquaria victoria. The incredible thing is, it makes its own fluorescent part, its fluorochrome, all by itself, just by folding correctly. No cofactors needed.
So you don't need to add any dyes.
Nope. And the real revolution came when scientists realized they could take the gene for GFP, clone it, and put it into other organisms.
So the cells make their own fluorescent tag.
Precisely. You can engineer cells or whole organisms to produce GFP, lighting them up from within.
And this led to a whole rainbow of fluorescent proteins, right? Blue, cyan, yellow, red.
Yep. Scientists tweaked the original GFP gene to create variants with different colors, improve brightness, better stability, a whole palette for biologists.
How are these used?
So many ways. One simple way is, as a reporter, you link the GFP gene sequence to the control region, the promoter of a gene you're interested in. So whenever that gene gets turned on in the cell also makes GFP. You get a direct visual readout of gene activity in living cells or organisms.
Like a little green light saying this gene is on here.
Exactly. Or you can add specific targeting signals, little molecular zip codes, to GFP to send it to particular organelles like mitochondria or the nucleus, lighting them up specifically.
Okay.
But maybe the most powerful application is creating GFP fusion proteins.
Meaning?
You genetically fuse the GFP gene directly to the gene of your protein of interest. So the cell produces your protein with GFP literally stuck onto it.
And it still functions, hopefully.
Often it does, yes. You have to check, of course. But if it works, You can now watch your specific protein, where it goes, how it moves, who it interacts with in a living cell in real time. It's arguably the best way to study protein dynamics in vivo.
So not just where, but what they're doing. How do you track interactions?
One really elegant method is FRAP, Förster resonance energy transfer.
Okay, FRET. How does that work?
You label two potentially interacting proteins with two different fluorescent proteins, a donor and an acceptor, with specific spectral properties.
Meaning their colors overlap in a certain way.
Right. If and only if the two proteins come very close together, like within 5 nanometers, basically touching the donor molecule when excited, can transfer its energy directly to the acceptor without emitting its own light.
So the donor's light disappears and the acceptor lights up instead.
Exactly. Or the ratio of their emissions changes. It's exquisitely sensitive to distance. So if you see FRET, you know those two proteins are interacting very closely. It's great for monitoring signaling events or the assembly of protein complexes.
That is clever. Like a molecular proximity sensor. What else?
There's also photoactivation, using special fluorescent proteins that can be switched on or change color with a pulse of specific light.
So you can highlight just a subset of molecules.
Yes, you can light up just the proteins in one part of the cell, for instance, and then track where they go over time, separately from any newly made proteins.
And you mentioned FRP earlier, fluorescence recovery after photobleaching.
Right, FRAP. That's different. You use a strong laser to deliberately bleach or destroy the fluorescence of GFP-tagged molecules in a small area of the cell.
Okay, so you create a dark spot.
Exactly. Then you just watch and measure how quickly unbleached fluorescent molecules from the surrounding area move into that bleached spot.
Ah, so it tells you how mobile the molecules are.
Precisely. You can measure diffusion rates, see if molecules are actively transported, or determine how long they stay bound to structures by how quickly the fluorescence recovers. It gives you quantitative dynamic information.
It really feels like spying on the cell, especially for tracking those fast, transient signals like calcium ions. That must be tough.
It is. Early methods used chemical dyes that change fluorescence when they bound calcium, for instance. But now, genetically encoded biosensors are often the way to go.
Based on fluorescent proteins again?
Usually, yes. They typically have two parts. A sensing domain that changes shape when it binds the molecule of interest, like calcium or KMP, or detects a pH change. And a reporting part, often two fluorescent proteins positioned just right for FRET.
So the shape change in the sensor alters the FRET signal between the reporters.
You got it. Binding the target molecule causes a conformational change, which changes the distance or orientation between the FRET pair, leading to a change in the color or intensity of the fluorescence you detect.
And you can design these for all sorts of things.
Yes. Calcium, CAMP, pH, kinase activity, even temperature. You can introduce the gene for the biosensor and watch signaling happen in real time in specific cells within a living organism. It's incredibly powerful for dissecting signaling pathways.
Okay, shifting focus a bit.
Right.
A big challenge with any conventional microscope, looking at thicker things like whole cells or tissues, is blur, right? Light from above and below the focal plane fuzzes up the image.
Absolutely. Out-of-focus blur is a major limitation. It obscures detail.
So how do you get around that to see a crisp slice?
There are two main strategies. One is computational, the other is optical.
Let's start with computational.
That's image deconvolution. It uses the knowledge of how the microscope optics blur a single point of light, that point spread function, or PSF we talked about. You collect a stack of images at different focal depths, which all contain blur from other planes. Then a computer algorithm uses the PSF to essentially reverse the blurring process mathematically, calculating where the light most likely originated from.
So it computationally removes the out-of-focus haze.
Exactly. It generates a set of clean optical sections. It's a bit like how a CT scanner reconstructs a 3D image from multiple X-ray projections.
Okay, so that's cleaning up the image after the fact. What's the optical approach?
That's confocal microscopy. It tackles the blur problem before the light hits the detector.
How?
Instead of illuminating the whole field of view, it uses a focused laser beam to illuminate only a single tiny spot in the specimen at a specific depth.
Okay, just one point at a time.
Right, and then crucially, there's a confocal pinhole aperture placed in front of the detector. This pinhole is positioned so that only light coming from that illuminated focal spot can pass through to the detector.
Ah, so light from above or below the focal spot, the out-of-focus light, hits the edges of the pinhole and gets blocked.
Precisely. It physically rejects most of the out-of-focus blur. Then the laser spot is scanned across the specimen, point by point, line by line, to build up a complete sharp optical section.
Very clever. And you stack these sections for a 3D view.
Yes, you can collect a stack of confocal sections to create a beautiful, high-resolution 3D image of complex structures, like neural networks or the elaborate internal structure of organelles.
So confocal versus deconvolution. When would you use which?
Good question. Confocal is often better for thicker, highly scattering specimens where there's a lot of blur to reject optically. It's also often simpler to implement. Deconvolution can be better for dimmer samples or situations where you want to minimize light exposure, as it collects all the light first, including the out-of-focus stuff, and sorts it out later. But deconvolution can struggle deeper into tissues, say beyond 40 or 50 microns.
And even confocal has limits for deep imaging.
Oh yes, scattering limits penetration. Usually you can't get much beyond maybe 150 micrometers with standard confocal. For going deeper into living tissues especially, you need multiphoton microscopy.
Multiphoton, how's that different?
Instead of exciting the fluorophore with one high-energy photon like blue or green light, you use two, or sometimes three, lower-energy photons arriving at the fluorophore at almost exactly the same instant.
Lower energy means longer wavelength, like infrared.
Exactly. And longer wavelength infrared light penetrates much deeper into tissue with less scattering. Plus, the excitation only happens right at the focal point where the photon density is incredibly high, so there's very little out-of-focus excitation or photo damage.
So deeper imaging, less background, less damage.
You got it. It allows imaging up to 0.5 millimeter, sometimes even more, deep into living tissues. Huge for neuroscience, developmental biology.
Incredible progress. But even with all this, we still bump up against that 200 nanometer diffraction limit for resolving fine details, right? Many things in the cell are much smaller.
We do. Ribosomes, viruses, cytoskeletal filament details, protein clusters, they're all smaller than the diffraction limit of light. Which is why super-resolution microscopy has been such an explosion in the last couple of decades.
Okay, let's dive into super resolution. How did they break that barrier? First up was SIM, structured illumination microscopy.
Right. SIM is one approach, typically getting down to about 100 nanometer resolution. It works by illuminating the sample not with uniform light, but with a striped or grid pattern of light.
Okay, patterned light. Why?
This known pattern interacts with the unknown fine details of the sample, creating interference patterns called moire fringes. These fringes are larger than the original fine details, large enough to be resolved by the microscope.
Like when you overlay two window screens and see bigger patterns.
Exactly like that. You take multiple images with the illumination pattern rotated and shifted, and then a computer algorithm analyzes how the sample generated those moire patterns to computationally reconstruct a super-resolved image.
Clever. And it works with standard dyes.
Mostly, yes. It's quite versatile, can do 3D, and works with many standard fluorescent labels.
What other ways are there to get super resolution?
Other methods play tricks with that plate spread function, the blurry spot from a single light source. One is STED, stimulated emission depletion microscopy.
STED. Sounds intense.
It is. The idea is to make the effective fluorescent spot much smaller. You use two lasers, one to excite fluorescence in a diffraction-limited spot, just like confocal, but then a second, very strong depletion laser, shaped like a donut, is overlaid on the first spot.
A donut beam.
Yeah, a ring of light. This donut beam is tuned to a wavelength that forces excited molecules back down to the ground state without emitting fluorescence. It depletes the signal. But because it's donut-shaped, it leaves a tiny hole of undepleted fluorescence right in the center.
So only molecules in the very middle of the donut hole get to glow?
Precisely. You effectively shrink the area from which you collect signal down to potentially 20 nanometers or even less. Then you scan this tiny effective spot.
Wow. Does that need special dyes?
Yes, it generally requires dyes that can handle this depletion process efficiently. But the resolution is fantastic, revealing structures like the details of nuclear pores.
20 nanometers with light. Okay, and then there's the single molecule approach, Paul and Storm?
Right. Single molecule localization microscopy, or SMLM. These include methods like PALM, photoactivated localization microscopy, and STORM, stochastic optical reconstruction microscopy. The core idea is completely different. It relies on a mathematical trick. If you image one single fluorescent molecule that's well separated from others, even though its image is a blurry diffraction-limited spot, the PSF, you can calculate the center of that blurry spot with much higher precision, potentially down to just a few nanometers if you collect enough photons from it.
Okay, find the center of the blue very accurately. But how do you image just one molecule at a time when they're packed together in the cell?
That's the genius. Use photo-switchable fluorescent proteins, or dyes molecules, that you can turn on and off with different colors of light.
Oh.
You start with almost all your labeled molecules in a dark or off state. Then you use a very weak pulse of activation light to randomly switch on only a sparse subset of molecules, so they're far apart from each other.
So only a few dots light up randomly.
Exactly. You image those few molecules, pinpoint their centers precisely, and then they either bleach or you switch them off. Then you repeat the process, activate another sparse subset, image them, locate them, turn them off.
Over and over again, thousands of times.
Tens or hundreds of thousands of times. Each cycle captures the coordinates of a few more molecules. You build up a list of millions of precise molecular coordinates. Then, you reconstruct the final super-resolution image by plotting all those coordinates.
That's incredible. It's like building the image molecule by molecule.
Pretty much. It has revolutionized light microscopy, allowing multicolor imaging, pushing towards live cell imaging, and revealing cellular structures at near molecular detail.
Okay, that's manipulating the light or the molecules, but what about manipulating the sample itself? Expansion microscopy, XXM? That sounds wild.
It's a totally different philosophy, and really clever. Instead of making the microscope better, you make the sample bigger.
How on earth do you do that uniformly?
You first fix and label your sample, just like for immunofluorescence. Then you embed it in a special kind of swellable polymer gel, like the stuff in baby diapers, but optimized. Critically, you chemically anchor your labels, or the molecules they're attached to, directly to the gel matrix.
Okay, labels locked into the gel.
Then you digest away the actual biological material, the proteins and lipids, leaving behind the labels embedded within this gel scaffold, like ornaments hung in space.
Wow.
Finally, you add water. The gel swells up isotropically, meaning equally in all directions, expanding maybe fourfold or even tenfold or more.
So everything just gets bigger but stays in the same relative position?
Exactly. Features that were originally, say, 70 nanometers apart, below the diffraction limit, might now be 280 nanometers apart after 4x expansion. Now you can easily resolve them with a standard confocal microscope.
That's genius. Effective super resolution without the fancy microscope.
You got it. You can get down to maybe 25 nanometer effective resolution. It's great for things like mapping RNA transcripts or tracing fine neuronal processes. And you can combine it with other techniques.
Okay, what about imaging really big things, like whole embryos developing over time or large chunks of brain tissue? Standard confocal or multiphoton might be too slow or cause too much photo damage for long-term imaging.
That's where light sheet microscopy, also called SPIM, selective plane illumination microscopy, has made a huge impact.
Light sheet, how does that work?
Instead of shining a focused beam through the objective lens, you use a separate cylindrical lens to create a thin sheet of light that illuminates the sample from the side, perpendicular to the detection objective.
So you only illuminate the single plane you're currently imaging.
Exactly. The detection objective looks at that illuminated plane. This has major advantages. First, you only excite fluorescence in the focal plane, dramatically reducing out-of-focus blur and improving contrast. Second, and crucially for live imaging, you expose the rest of the sample to much less light, minimizing photobleaching and photo damage.
So it's faster and gentler.
Much faster and much gentler. You can acquire entire 3D volumes very rapidly, hundreds of image planes per second, making it ideal for watching dynamic processes in whole embryos over hours or days, and for rapidly imaging large cleared tissue samples like entire mouse brains to trace neuronal circuits.
Amazing. One more light technique. What if you want to see single molecules right at the cell surface, but the background fluorescence from inside the cell is drowning them out?
For that, there's TIRF microscopy, total internal reflection fluorescence.
2RF. How does that eliminate background?
It uses a neat optical trick based on how light behaves at an interface, like between the glass cover slip and the watery cell medium. You shine the excitation laser beam through the objective lens at a very specific shallow angle so that it hits the glass-water interface at greater than the critical angle. This causes the light to undergo total internal reflection. It basically bounces off the interface instead of passing into the sample.
So no light goes into the cell. How do you excite anything?
Well, not quite no light. The reflection creates what's called an evanescent field. It's an electromagnetic field that penetrates a very short distance, typically only 100 to 200 nanometers beyond the glass surface into the sample.
So only molecules extremely close to the cover slip get excited.
Exactly. Anything deeper in the cell is effectively invisible, because the evanescent field doesn't reach it. This gives you incredible signal-to-noise for imaging processes happening right at the plasma membrane, like vesicles docking and fusing, motor proteins walking along filaments near the surface, or the assembly of adhesion complexes.
But it's limited to that very shallow surface layer.
That's the main limitation, yes.
Yeah.
Perfect for the surface, but can't see deep inside.
Wow. That's an incredible toolkit just within light microscopy.
Right.
But sometimes you just need higher resolution, right? Down to the molecular level.
That's when you have to leave light behind and turn to electrons. Electron microscopy, or EM.
Using electrons instead of photons.
Right. Electrons have much, much shorter wavelengths than visible light, which allows for vastly superior resolution.
But there are big trade-offs you mentioned.
Major trade-offs. Generally, you're looking at fixed, dead cells because the sample has to be in a vacuum and withstand an electron beam. Specimen preparation is much more complex and artifact-prone. You gain resolution, but potentially lose some lifelike context or dynamics.
How much better is the resolution, typically?
The wavelength of electrons used in microscopes is incredibly short, like 0.004 nanometers. Theoretically, resolution could be near atomic. In practice, for biological samples, due to lens aberrations and sample issues, it's more like 1 nanometer or 10 angstroms.
Still 1 nanometer. That's 200 times better than the 200 nanometer limit of conventional light microscopy.
A huge leap in resolving power. You can see the detailed shapes of organelles, macromolecular assemblies, viruses, and newer electron sources, like field emission guns, push that resolution even further for some applications.
So what's the basic setup? A transmission electron microscope, TEM.
The TEM is kind of analogous to a standard light microscope, but scaled up and flipped. You have an electron gun at the top generating the beam, magnetic coils acting as lenses to focus a beam, the specimen stage, and then projection lenses forming an image on a detector at the bottom.
And it all has to be under high vacuum.
Yes, electrons scatter off air molecules, so the entire column has to be evacuated, which is a big part of why sample preparation is so challenging.
Right. How do you prepare squishy, watery biological samples for a vacuum and electron bombardment?
Traditionally, it involves harsh steps. Chemical fixation, usually with glutaraldehyde and osmium tetroxide, which also acts as a stain. Then dehydration, replacing all the water with organic solvents like ethanol or acetone. Then infiltrating with liquid plastic resin that hardens. And then slicing that block into ultra-thin sections.
Ultra-thin meaning?
Really thin, like 25 to 100 nanometers thick, way thinner than light microscopy sections, because electrons don't penetrate very far. You need a special ultramicrotome with a diamond knife to cut these.
Wow. That sounds like it could easily introduce artifacts.
It absolutely can.
Oh.
Which is why rapid freezing, or cryofixation, is considered the gold standard for preserving structure most faithfully.
Freezing so fast that ice crystals don't form.
Exactly. You plunge the sample into a cryogen like liquid ethane, cooled by liquid nitrogen, achieving cooling rates of thousands of degrees per second. Water vitrifies, becomes a glass-like solid without forming damaging crystals.
And then you can section it while it's still frozen.
You can do cryosectioning, yes, or examine the frozen sample directly in some cases, cryo-EM, or use techniques like freeze fracture or freeze substitution, where you replace the vitreous ice with resin at low temperature. It preserves structures much closer to their native state.
Can you label specific proteins in EM, like immunofluorescence and light?
Yes, using immunogold labeling. Instead of a fluorescent tag, the secondary antibody is linked to a tiny, electron-dense colloidal gold particle.
So you see little black dots in the EM image where your protein is?
Exactly. The gold is very dense, so it scatters electrons strongly and shows up as a distinct black dot. You can even use different sizes of gold particles to label multiple proteins simultaneously.
Okay, so contrast in TEM comes from differences in how much electrons are scattered. Biological stuff is mostly light atoms. How do you get enough contrast? You mentioned osmium.
Right, osmium tetroxide fixes and stains lipids and proteins. But usually sections are also stained with solutions of other heavy metal salts like uranium, uranyl acetate, and lead citrate. These heavy atoms bind non-specifically to various cellular components, increasing their electron density and making them visible against the background.
So it's basically heavy metal staining.
Pretty much. Different structures pick up the stains differently, giving you the characteristic EM contrast.
Are there other ways to get contrast, especially for isolated molecules?
Yes, two main ones. Shadowing involves laying isolated molecules like DNA strands or viruses on a flat grid and then spraying them with a thin layer of heavy metal like platinum from an angle.
Creating little shadows?
Exactly. The metal coats the surfaces facing the spray, leaving a shadow behind the molecule. It gives a nice surface rendering, but resolution is limited by the graininess of the metal coat, maybe 2 nanometers.
And the other method?
Negative staining. This is great for seeing the shape and subunit structure of isolated particles, like viruses or ribosomes. You mix your sample with heavy metal salt solution, like uranyl acetate, on the grid. The solution dries down, forming a dense electron scattering background everywhere except where the particle is sitting.
So the particle excludes the stain.
Right. The particle itself is unstained and appears bright electron-lucent against a dark background of heavy metal, hence negative staining.
Okay, TEM gives amazing internal detail, but what if you want that 3D surface view, like the outside of a cell or even an insect?
For that, you use scanning electron microscopy, SEM.
SIM. How is it different from TEM?
Instead of transmitting electrons through a thin section, SEM scans a focused beam of electrons across the surface of a bulk 3D specimen.
And detects.
It detects electrons that are scattered off the surface, backscattered electrons, or electrons that are knocked out of the specimen's atoms by the beam, secondary electrons. The intensity of these signals varies depending on the surface topography.
And that gives the 3D look.
Yes, SEM images have a fantastic depth of field, so you get that really striking three-dimensional appearance, with highlights and shadows revealing surface texture. You usually coat the sample with a thin layer of conductive metal, like gold, to prevent charge buildup and enhance signal.
What kind of samples work well?
It's great for whole cells, tissues, small organisms, things where you want to see the overall surface architecture. Resolution is typically lower than TEM, maybe 1 to 10 nanometers, but modern high-resolution SEMs are closing that gap for some applications.
But again, TEM sections are 2D projections, losing 3D info. How do you get true 3D reconstructions at EM resolution?
Electron tomography. It's analogous to a medical CT scan, but performed inside the TEM.
How does that work?
You take your specimen, usually a slightly thicker section than normal TEM, maybe 100, 300 millimeter, put it on a special holder that can tilt, and then acquire a series of images as you tilt the specimen through a wide range of angles, maybe from plus 60 to negative 60 degrees.
So lots of 2D views from different angles.
Exactly. Then, computational algorithms, similar to CT reconstruction, combine all those tilted views to calculate a 3D reconstruction, a tomogram, of that volume of the cell.
Giving you the 3D architecture.
Yes. And you can computationally slice through that tomogram in any direction. Even more powerfully, if you have multiple copies of the same structure within the tomogram, like ribosomes or proteasomes, you can computationally extract them, align them, and average them together.
Subtomogram averaging.
Right. This averaging boosts the signal and can achieve resolutions of 1 to 2 nanometers, letting you determine the structure of molecular machines within their native cellular environment. It's a crucial link between cell biology and structural biology.
Which brings us to the absolute cutting edge for structure. Cryo-electron microscopy, cryo-EM, particularly for single particles. You mentioned this briefly. How does it achieve near atomic resolution?
Cryo-EM single particle analysis has been revolutionary, truly deserving its Nobel Prize. The challenge was always imaging unstained macromolecules. They offer very little contrast, and the electron dose needed for high resolution would destroy them.
So how did they solve it?
2 key breakthroughs. First, sample preparation. You take a purified sample of your macromolecule, like a protein complex or virus, apply a tiny drop to an EM grid, blot away most of it to leave a very thin film, and then plunge freeze it incredibly fast into liquid ethane.
Vitreous ice again, preserving the structure without crystals.
Exactly. The molecules are trapped in random orientations within this thin layer of non-crystalline ice. You keep the grid frozen at liquid nitrogen temperature inside the microscope.
And the second breakthrough?
Detectors and computation. New direct electron detectors are much more sensitive and faster than old film or CCD cameras. And sophisticated image processing software was developed.
How does the process work?
You use the TEM to take thousands or even hundreds of thousands of very low-dose images of these frozen, randomly oriented particles. Each image is extremely noisy.
Low dose to avoid damage.
Right. Then the software goes to work. It identifies the individual particle images, computationally determines their different orientations, sorts them into classes based on viewpoint, aligns the images within each class, and then averages them together.
Averaging thousands of noisy images cancels out the noise.
Precisely. It dramatically increases the signal-to-noise ratio. Finally, these average 2D projections from different angles are combined computationally to reconstruct a high-resolution 3D density map of the molecule.
And the resolution now is?
It's routinely reaching 0.2 to 0.3 nanometers, 2 to 3 angstroms, which is well into the atomic resolution range, comparable to X-ray crystallography. You can often build an atomic model directly into the cryo-EM map.
What are the advantages over crystallography?
Huge advantages. You don't need to crystallize the protein, which is often the bottleneck. It works for very large, complex, or flexible assemblies that resist crystallization. You can capture different functional states or conformations by analyzing subsets of the particles, and you need much less sample.
So it's replacing crystallography.
Not replacing, but complementing it beautifully. Sometimes you can solve the structure of subunits by crystallography and then fit them into a lower-resolution cryo-EM map of the whole complex. They work together.
This really bridges scales, which leads perfectly into the last technique, correlative light and electron microscopy, CL-EM, combining the strengths of both.
Exactly. Fluorescence microscopy tells you what molecules where, functional information, specific labels, but with limited resolution and context. EM gives you fantastic high-resolution context, ultrastructure, but it's hard to identify specific unlabeled molecules. CL-EM aims to combine both on the same sample.
How do you do that?
One way is to image your sample first with fluorescence microscopy, maybe live cell imaging, or immunofluorescence on fixed cells to find your event or molecule of interest. You record its coordinates precisely. Then, you process that exact same sample for EM, relocate the same area, and acquire EM images or a tomogram.
And overlay the fluorescence signal onto the EM structure.
Yes. You correlate the fluorescent spot with the underlying ultrastructure seen in the EM. That green dot corresponds to this specific vesicle. It requires careful sample handling and finding the same spot again.
Sounds challenging. Is there another way?
A more integrated approach, especially for high-resolution correlation, involves techniques like FIBSEM, focused ion beam SEM, which we mentioned.
The ion beam milling machine.
Right. You might first do super-resolution fluorescence microscopy, like PalmStorm, on your frozen sample to precisely locate molecules. Then you put it in the FIBSEM. The ion beam mills away a very thin layer, maybe 5–10 nanometer from the surface of the frozen block. Then the SEM images the newly exposed face. Mill image, mill image.
Building up a 3D volume, slice by slice.
Exactly. You generate a high-resolution 3D EM volume of the cell, and because you know the original fluorescence coordinates, you can precisely map those fluorescent signals onto the 3D ultrastructure. It gives incredibly precise correlation, down to maybe 5 nanometer accuracy. It's very powerful, but technically demanding.
What an amazing array of tools. It really feels like we've journeyed from fuzzy blobs to seeing atoms.
It's been an incredible progression, hasn't it? The history of cell biology is so tightly linked to the history of being able to see smaller and smaller things more clearly and more dynamically.
So to wrap up, this whole deep dive highlights that central theme again, trade-offs.
Absolutely. Every single technique we discussed involves compromises. Want high resolution? It probably means more complex sample prep, maybe no live cells, higher cost. Want live cell dynamics? You might sacrifice some resolution or can only look near the surface. Want specific molecular identity? You label it, but then the unlabeled context might be hidden.
It's always a balance. Contrast, resolution, signal to noise, speed, how deep you can see, how much damage you cause. Improving one often means sacrificing another.
Understanding those trade-offs is key. It's what allows researchers to choose the right microscope, the right technique to answer their specific biological question effectively. There's no single best microscope. It's always about the best tool for the job.
A fascinating perspective. It makes you think, as these tools continue to improve, pushing boundaries despite the trade-offs, what new aspects of life will come into view?
Exactly. What questions are we not even asking yet, simply because we haven't had the tools to visualize the answers? The constant innovation in microscopy keeps redefining what's possible, and that's what keeps driving discovery in biology.
Well, that brings us to the end of this deep dive into the incredible world of cellular visualization. We really hope you feel much more informed about the amazing toolkit scientists have at their disposal.
Thank you for joining us. We hope you found this look inside the cell as fascinating as we do.
Indeed. Thanks for listening to the deep dive. We hope you'll join us next time.